Hello, g'day, good evening to wherever you are and sit. Hello and welcome. Just like Vin, hello Vin and Aussie Autos FM. G'day, g'day. How are you? So today is actually World Cannabis Day. I heard on the radio before. So yes, World Cancer Day, not cannabis. Sorry, it's World Cancer Day. So I thought it might be relevant to go through some slides of anti-cancer processes in the way that how cannabinoids help kill and get rid of cancer or keep it at ease and stuff like that. So I'll wait for people to chime in. How you going, Dave? 1969, I can show that. Big Dave, big Aussie Dave's here as well. Wow. G'day, fellas. How's it going? Today was going to be an open topic, but we can have open topic too. And it's World Cancer Day. So I was going to show some slides on how cannabinoids inhibit the cell cycle, getting rid of it, keeping all the anti-cancer cells at rest and keeping them at bay. Hey, PTP, how you going, mate? All right, well, I'll give them a little thingo. Lene, how are you going, Lene? Cheers. So if you've got any open medical cannabis questions, you're welcome to ask. And I'm going to talk a little bit about anti-cannabinoids, anti-cancer cannabinoids. There's only a little bit, but there's a couple of slides that are showed relevant because today is World Cancer Day. So sorry to those poor people that get cancer. And if you have got cancer, you can think about using your THC and CBD blended as a way to inhibit or kill cells of cancer. And I'll show some slides in a minute. PTP, there you go. Me, there's Monty. G'day, Monty. How are you? How's it going, Monty? All right, well, save your questions a bit by the look of it. I'm going to show some slides on some anti-cancer stuff. I'm a cancer, there you go, already. He's a cancer survivor, see, and he uses the THC and the other cannabinoids to his advantage. Well done, Monty. Sorry you had it, mate. I've had close family have it as well. It's not very good. Hit the like button. Oh, Terrence, how you going, Terrence? You reckon hit the like button? Good evening, good peeps. Hello, GMF. Good Genetic Memory Farms. I hope you all are safe and stoned, he says. Yes, I'm going to talk. It's World Cancer Day, mate. So I'm going to talk a bit on some anti-cancer cannabinoids and the pathways and processes in which others can do it and reap the rewards from it. So I'll get back into it. Present. Here we go. Slides. Your computer. No, wrong button. Share screen, that's right. Woohoo! Good medicine. Allow. Okay, so this is a study done, cancer studies done with cannabinoids, that they had a combination, they called it CM, which is a cannabinoid mixture, and they found that the cannabinoid mixture, which contained all these different phytocannabinoids, CBD, CBG, CBN, CBC, and there's quite a few more, and the THCs, they found that when they were administered, that exposure resulted in both the totals of the reactive oxygen species and provoked mitochondrial membrane potential inhibition. So that stops the bad reactive oxygen species are the enzymes, peroxidome, Geez, I can't think of it now. I shouldn't have had a good session before. But the active oxygen species, it inhibits them anyway. CM-induced apoptosis is when the cell kills itself. So this cannabinoid mixture induced cell death of oral cancer cells, suggesting that cannabinoid mixtures at lower concentration significantly enhanced capase activity in that gene. Was another interesting thing too. CM had a statistically significant dose-dependent growth inhibitory effect on those genes, which supports oral cancer. Induced apoptosis. Okay. CM exposed results to reactive oxygen. All right. Well. All right. That's a study that they found that's true. There's another thing in here. This is the pathways from how they work. So this is the cell wall, cell membrane. That's the apoplast out there, and inside here's the cytoplasm, or inside of the cell. So this is in the cell wall, and these are the different receptors that are there. So you CB1, CB2, TPRV1, and your GPR55s, and CBD and THC activate different ones. So by those molecules coming in, as you see in the colors, they'll go through and activate it, which will set off the things at the other end, which sets off these pathways. And pathways end at cell cycle at arresting it, so it'll kill it. It'll like stop it and hold its growth stage. That's what arrest means. Autophagy means when it eats itself, so it'll turn it and sort of start and eat itself, and it'll die. And apoptosis is cell death, so it'll start and break up. So there's how to do it. And the main anti-cancer pathways. Cannabinoids inhibit cell cycle progression. I'm not going to read out them. They can induce apoptosis, cell death, or autophagy, which is the cell eating itself by activation of cannabinoid receptors CB1 or CB2, but can also be induced by mechanisms independent of CB1 and CB2. So the combination of your THC and your CBD does this. So if you've got cancer, really, truly, this was in the molecular mechanism of cannabinoids in cannabis progression. That's the heading, and it was found in 2021. And the book or the paper is the International Journal of Molecular Science. So that's rad. I should get back to chat. Actually, there's cannabinoid therapeutics in orofacial pain management. Oro, that's pain in the face. Oro, in the throat. This is a study done in, hey, look at that, James Cook University in Queensland, and they found that it works, that the cannabinoids actually do work in dental medicine. And here's some other pain. Cannabinoids are the best option for chronic neurological pain, and if you've got cancer and stuff like that, it might be the same sort of path. So this chart shows all the different type of opioids, or the things that you can get when you have cancer, and it shows that over on the right here, there's different pain relief, quality of life, and a different dizziness, drowsiness, all types of different effects, and they color charted them. And then over on the left, they've got the degrees. So I'm only interested in the top two. So pain relief and quality of life. So if you go for pain relief, they're all pretty good. Ibuprofen shit, don't use it. Actually, I'm not a doctor. So, and the quality of life's more so. So look at the yellow. So yellow with TB, with the RSO, or the combined combination of your medical cannabis, massive. THC, massive. Everything else, relatively low, let alone the side effects. So they haven't gone into the side effects yet, but yeah, that's very interesting. All right, that'll do. Change that. Go say, catch up with the chat. Nice. I didn't know it's World Cancer Day. Yep, smoke up all your cannabinoids. Yes, for those who have cancer, it will help. Or there's in Canada, when I lived in Canada for 5 years, that over there, they have 2 cases of this, my mate's wife had cancer, and as soon as they went off their RSO, her cells used to grow and it used to advance, so she'd get back onto it. They'd make some more again, and then it would keep it at ease. And then a friend had a dog, and with his dog too, he said the same thing. He said, I just give it to my dog, and then as soon as I didn't give it to it, you see the lumps just getting bigger and bigger, but you just give it to him and it just stays the same. And it was a big cyst from cancer. So there were two experiences where people have said that they worked with them, as well as the studies. So that's pretty cool. Whoops. Cold, definitely. I'm just reading the chat a bit. If you've got any questions too, it's an open discussion, open medical cannabis topics, because today's World Cancer Day, so I thought I'd just throw in those slides. That's really all I've got to do, touch on that subject. Banana Farmer, how you going, mate? G'day. Terrence, that's funny. Looks like you're writing from a different country. AussyCC, oh yeah. Do you believe cannabis sometimes has apomixis tendencies? Apomixis? APOMIXIS. I'll just Google that. APOMIXIS. Apomixis. What is apomixis? Apomixis in flowering plants is defined as the asexual formation of a seed from the maternal tissue of the ovule, avoiding the process of meiosis and fertilization. Oh, okay. What was the question again? Do you believe cannabis sometimes has? Yes, it definitely does, mate. That's sometimes when, that's a way that it forms the, I'll bring up a slide for you. It forms tetraploids and triploids. Where am I looking in? Soil science. We're going to go into seeds, I presume. I think it's in genetics. I'll just share the screen so you can have a bit of a look, see what I'm doing. Keep it real. Share screen. Share seeds. There's no chart there that I wanted to see. I wanted to see the germination of the seeds, and then I can explain to you in it what's going on and how the triploids form because of the different cells. There's egg cells that are in the middle, down the bottom. Then you've got your mother cell in the center, which has different N ratios, which is your, hang on, I'm just trying to do two things at once. It's not in seeds. It's got to be in genetics then. It's only just one chart, come on. I'm trying here for you, Banana Farmer. Genetics is humongous. Are you still watching? Yeah. Jesus, triploids. Maybe in here. No, shit. Come on, it's not hard. Fertilization, seed, embryogenesis. It's got to be in here. Yeah, there it is. Sweet. Yep, sharing. Oh, that's full on. Yeah, this is it. So this is how, so the pollen grain will sit on top of, this is the anther or the pistil. So it'll land and sit on top of here, and then it'll travel all the way down. It puts out little calcium channels and it gets a signal, so it knows it's going down. And then it has its two sperm cells that are following the calcium channel, the vegetative nucleus, and it'll go into the ovary, and then in the center has its central cells, and then the bottom is its egg cells. And in here, the mother's central cells, they've got a 2N ratio, so the amount of chromosomes that they've got is already 2, and down here it's only 1. So if it actually gets through to the center, that's where it happens, and that's one of the ways that you can get triploids. It's called double fertilization, as you see, but yeah, that's how, one of the ways it happens. Cool. Is there anything else in here that's interesting? Pollen tube germination. This is a stain under a microscope, and you can see how the pollen tube actually goes and germinates. So in this one, it starts up top and then it'll slowly put the cell down. So it'll put down that big long thing that you saw before to try and curve around and reach up through the micropyle into the ovary. Self-pollination cross. Self-incompatibility, no. Monoecious plants and dioecious. Cannabis can be both. It's rated as dioecious, but it can be actually both. The embryo sac, here's a bit of a zoomed in. So that's when that pollen tube that we saw is highlighted before, it'll come around in through the micropyle here and try and reach into the ovary and hit one of the cells. And sometimes that, what you said before, what was that word you said? APOP something, that can occur, and that's when it'll go to one of the other ones. And you get your natural forming triploids. Anything else in here that's interesting? I like genetics. See, there's your one ends, and then the middle one has two ends. So these all have one end, and then these will have two sets of chromosomes. So that's how you get your triploids. That's inside of the pollen grain. So there's one end as well. So it can only, so one end plus one end equals two end, which gives you two sets of chromosomes, which is the normal diploids. But if you want a triploid or a tetraploid, you got to go and do this. This can be what Mother Nature's way of doing it, or you can go and induce it in ways that I've done it. That's how I produce it. That'll do. We don't want to get into that. Too heavy. We'll keep going down. All right. Thanks for your question, Banana Farmer. That was good. Apomixis tendencies. So yes, it does have it from inducing it with a few cool things, and it happens in, that's one of its processes in nature, how it happens. There's many different other ways how it occurs too. It's really good. That should really do a section on polyploids and triploids. Polyploids is a definition to mean that any more than it's ploidy. So its ploidy is its measure of its chromosomes, and then poly means more or many of. So that means it's how many sets of chromosomes it's got. So if it's diploids 2, triploids 3, and then tetraploids 4, and then it goes up from there. Like strawberries, strawberries 8, it's octoploid. What's hexaploid, which is 6? Don't know. Anyway, back to it. I'll keep cruising down. Good afternoon. Yeti, Mountain Trading. How are you, Yeti? Good afternoon, good afternoon. Jeff Papalia, how are you, mate? Nice to see you. Main Green Grower. Oh, gee, Main Green Grower 420. He's in the freezing north. Where is Shatter on that graph? On that Medical Cannabis graph was up before. Yeah, Shatter would be highly up there. It'd be on the THC. He means this one. Share screen. This one. So that's the, he said, where's Shatter? So this would be cannabis flower. So if you were to condense it, which is what Shatter is, that would be just, yeah, the winner by far, because you can see this is the winner by far here. The combination's up around, what, 79, and the next pill was 60 or even 58. So yeah, it would win by far. Shatter, well done. We don't have much of that in Australia. I don't think it's medicinally released yet, mate. It's crazy. At least they're getting to it. They're trying to start here in Australia. Good suggestion, though. I also believe, says Lene, cannabis prevents cancer and tumour growth. Yes, bless Mother Nature's medicine. Yes, it halts it. It does that with the cells, like we were talking before, with the apoptosis, how it can cause cell death from bad cells. Big Aussie Dave reckons, after my wisdom teeth, they told me I couldn't smoke for a week because sucking will cause dry socket. So I used a bucket bong and let it just waft into my mouth. Yes, they were true by saying that, and but good on you. See, there's always alternatives for cannabis. You could have eaten it as well. That's a good way. Or maybe chewed on some lollies while you had your bongs or your vaporizer. So it kept because when you chew, it releases the enzymes in your mouth, which is the myrmidase, it's called, and that's produces all the saliva and gets it all water, and that's a enzyme that helps break down all the stuff. So yes, if you chew it, that's why when I have my protein shakes, I chew, because it produces that natural enzyme that helps it break down. But yeah, good on you, Big Aussie Dave. He still found a way to get his relief. Is the question. Big Aussie Dave, like a female plant suddenly producing seeds on its own. That's a stress tendency, and the more stress that the plant's under, the higher. I spoke a bit this last week on the homozygosity and heterozygosity. So when the parents are breeding, if you've got two very distant parents, I'm not even going to say actually, because if you've got two distant parents, it'll breed up like an Asian and an African. You'll get a lot of different traits, and they won't be very stable. But if you were to get a stable breed and breed it back with itself or something like that, that's when its homozygosity gets very stable, and that's how to stabilize genetics. So most of the seeds that are put out these days aren't stabilized, so you'll find that they're in their first or second generation that they've been bred with the parent, so they're not stable. So they've got a very high herm tendency, you could say. So that's probably to induce that ham tendency, you've got to stress it under some sort of abiotic or biotic stresses, and that's how it happens, I suppose. Or you can get genetics which will have like a dodgy FL1 gene, florigen gene, which won't produce the flowering protein properly. So you can get different mutant genetics traits also that can do the same thing that you're talking about, mate. So it's not just from stresses. It's more so from stresses, but because of the high amount of mutant varieties that there are out now, that can also happen. Because the plant's got 35,000 genes, so any one of those can mutate, and if it's not bred correctly and properly, it can have this happen. Because Mikes, how you going, Mikes? It's the way. Intersexing is a real thing. Yes. RSO is so beneficial, helps me with numerous issues. Yes. Rick Simpson oil, the combination of the extraction of the cannabinoid flower, cannabis flower. Jesus, it was good this morning, wasn't it? So yeah, it really does work. World Cancer Day is today. All those poor people who have cancer. We all know someone. There's that many. Oh, there. Apomixis on the side. Apomixis. How you going, Critter? Says good morning. Nico D, long time no see. Yes, hello, my friend over there. Hope you're doing well in your yes, your climate. It's freezing where you are at the moment. You be doing well though. He always is, Nico. Critter. Nico, today's a open medical cannabis topic, so if you've got any questions on medical cannabis, feel free. I was talking about anti-cancer cannabinoids because today's World Cancer Day. I was showing that graphs and those slides earlier on. But it sort of got into that's it for those slides. So yeah, if anybody's got any open medical questions, cannabis questions. Morning. I was scrolling down here. Is there any science behind stressing the plant shortly before harvest to pump yields, or is it just theoretical? Says Big Aussie Dave. Is there any science behind? All right, what can I show you? Well, I can tell you actually that stressing a plant will activate its different pathways. So most of the pathways that I have not explained that. Let's bring up some charts. So what chart should I be going to here? Stressing plant deficiencies. Yes, let's try in here. All right, let's see what I got in here. I'm just going to I don't know what's in here, but I'll share it. Where are we? Entire screen. Plant deficiencies. There's not many of them. And so I don't see the chart that I was going to look for. Oh, what was the question again? Is there any science behind stressing the plant shortly before harvest to pump yields? I can answer that. I didn't read it right. I'm so bad with English, sorry. It's yields, no. The answer is definitely no. Shortly before harvest, because yields is to do with plant tissue, and that's to do with the amount of growth that you've got. So a plant cell reproduces itself about every 15 to 16 hours. So yield takes weeks to happen. So that's a long-term process. But if you're asking, I read the question as asking about, does it increase the potency? So I was thinking about potency, and I was thinking, I think that's what you might mean. So does it bump up the numbers? So does it get your THC, CBN, all those numbers? Can you increase them by harvesting, doing anything before harvest? Like people have put pins and nails through stems. They've done other things to stress it out. And I want to show you some charts relating to that. I'm still sharing the screen, aren't I? Yeah, I am. No, that's not the one. That's not the one either. This one. Oh yeah, I was going to see what I got in here, see if I can have something related to it. Cation exchange. No, definitely not. Leaching requirements. The amount of water needed to remove excess salts. No. Did you know that Tilray actually said that if you hang your plants upside down, their scientists said they hang their plants upside down so that it increases their runoff, so the cannabinoids can get higher. That's your essential plant nutrients, 17 of them. 3 of them come from the air, carbon, hydrogen, and oxygen, 95% of them too. Deficiency chart, no, it's not going to help. Nitrogen cycle, no. Then we're going to get into deficiencies. So by stressing it, what you're going to do is you're going to activate different pathways, because that's what I wanted to find, any different molecular pathways. I was hoping I had some signaling ones. And from doing that, it's going to slow the growth a bit because it's going to stop it and it's going to fend for itself. So it'll activate its immune system. So, oh yeah, see, like here, this is when I did an endophyte removal of a cultivar. So this had Pseudomonas syringae was the name of the bacteria. And I did a process of removing its endophytes and putting new ones into it. And this is the same cultivar on the right-hand side. So the left where it's sick and the right when it's fixed. So what's happening, if you're stressing it at the end, it's going to activate its pathways and want to always try and help itself. Like when we're sick, we haven't got much energy. Same thing with the plant, it activates its immune system. So if you stress it out, it'll activate that, and so it won't finish up properly, it won't finish strong, those types of things. But you asked for any science behind it. I can only speak from experience with this one because I looked for some charts, sorry. And another way is the black, when you harvest, do you keep it in the dark for 24, 48 or 72 hours? 2 people have done the test where they've got the individual plants tested through labs, and they both found that by 48 hours after darkness that they got a couple of % higher on their THC. I did it a couple of times and I was very happy with the results too, but it takes up space and it takes up days. So it depends on your setup, your plant counts, etc., and stuff like that. What else we got in this deficiencies? It might be at the end. Oh, there you go. Go on, Pseudomonas syringae. Oh, that's, you can see the new growth. So here, see that's old growth. So that was what's happening then too. Well, so here it's got its, say for instance, it's got a problem, and it's just woody and it's aged. They're sclerenchyma cells, they're called on the outside of the plant. So that's just aged and got a little bit of bark on it. But you can see that the new growth here is very fast. So this would have been when there was no pathogen problems, and here is when there is a problem. So you can see when the problem was ridded, when it was gone, it just took off, and it hasn't had any time to put a bark layer on yet. So it's a boron, yeah, good. Did you know boron, actually it's such a low, no, it's molybdenum I was thinking of. Yeah, his Pseudomonas syringae that I had pulled out of the other plant, I did some tests on it as well. It was such a good experiment, that one, removing pathogens. If you've got a really good cultivar you want to keep, you just, yeah, tissue culture, because Pseudomonas syringae, yuck, look at it. Ugh. There you go. There's a cannabis with Pseudomonas syringae, early stages. I thought, what sort of chlorosis virus is here to this? And then looked into it further and thought, oh, you're kidding. Not very good, mate. But there's a way to get rid of it, though. It's worked very well. Look at how sick that is. Yuck. That was because, yeah, this happened from outside getting sprayed with poison on the ground. Some of the travelled and got through into the plant, and then it spread. So it went, and that's through allelopathy. So it'll come in through one tissue, it'll go systemic, it'll produce a methyl signal, and then it goes into the air, and then the other plants pick it up, and yeah, unfortunately, it spread like wildfire. But you can get rid of it. There's chlorosis virus. Yes, cannabis chlorosis virus. I think I'm a bit off track here. I better get back to it. What is chlorosis virus? Nitrogen deficiency. All right, anyway, we're out of it. I hope that's... You asked for some science, though, and I couldn't give you any science, which is charts and things. I'm sorry, Big Aussie Dave, I'll let you down, mate. Great question, though. There might be some shining to the light beneath it. I better read down. Cheers. Ginseng would be a hexaploid. Oh, there you go. There's an example of a hexaploid. Thank you, Yeti Mountain Trading. Poison ivy's a hexaploid too. Oh, heaps of hexaploids. Mother Nature does it when it comes to an event, when there's like an ice age or something like that. It's the plant can go and produce and go into these traits, like tobacco is a polyploid naturally. What else is, what's that list I'm trying to picture? Oh, well, it's not a tetraploid thing. What I'm talking about. Back to it. A power hitter. Works well for dentists as well. You can add it, Monty says, you add a bit of pineapple juice to your protein shake, it helps break down, digest quicker. Okay. Cloudscape Mystico, how are you going, mate? Cloudscape checking in. Free Ted K. Whatever that means. No, it's not political here, mate. Oh, GMF. He's got a question. He's got a medical question. For the room, I have a friend in hospital, I think. He likes to smoke, but what kind of input does anybody have for things for him to try? Strains, preparations, anything. I have a friend in hospital, in hospice. I think it's hospital. He likes to smoke. What kind of inputs and strains? It depends what he likes. If he gets anxiety, I'm caffeine sensitive, so if people give me sativa, I'll be binging around the room and I won't be able to sit. So that would be shit for me, as an example. So indica, I'd suggest, because it agrees with nearly everybody. And yeah, if he's in hospital, he can't really smoke. So maybe some edibles for him or some oil might be good. Yeah, that's what I suggest. What does everybody else think? Because this is a broad question for everybody. I'll leave it up. Big Aussie Day said, very high THC, maybe an indica. Just what I said. That's a contrast. Thanks, Dave. I was going to flower an Afghani for him next. Yes, well done, GMF. Very high indica. I see they're just indica. They're not even indica dominant. It comes from the indica, in the region where they're named from, in the Hindu Kush mountains. Hey, Family Falk, how you going? Just it's an open medical cannabis topic today, Family Falk. I was discussing anti-cancer cannabinoids early and showing some slides for that. So we've sort of gone around a bit of that, on that. We talked a bit on apo mixes and how the ploidy was happening in the plants and how it naturally happens, how you can naturally have the triploids, the tetraploids in cannabis. I've been doing experiments for 2 years on tetraploids, and it's pretty cool. Very cool. All right, I'm reading down here. Yeah, the darkness period. I was wondering about that, he says. Yeah, if you've got time, try it by leaving it in 30, probably the two people I spoke to, how they've gone to lab tests, they said that they did it for 48 hours. They found it works best. And they did the 24, 48, and 72-hour tests. And you can see on the charts that it went up a little bit in the 48-hour 2 times. So, and yeah, something interesting. Post-harvest techniques. I did a course last semester. It was called Post-Harvest Operations of Agricultural and Cereal Crops. And there was some really cool things on drying, the ways that we can dry it quicker and better. Because did you know they had in California, some of the people might have heard of this in the States, there was a competition recently held where all of the samples that were held got retested, and they come up with too high in fungus and too high in bacterial counts. So it found that they couldn't be put in, and the show was called off. So, where was I going to? Shit. Oh, no. Yeah, the darkness, I was wondering about that. Oh, I had a good point to make about that. So if they'd done something differently, the outcome would have been different. That's the point I was trying to get to. Oh, well, sorry about it. Geez, that's a shame. Oh, well, I'll just keep reading down. So potency can be increased, but not heavy yield or weight in that short period. Yes, Big Aussie Dave, because the cells, for them to grow, it takes 15 to 16 hours in tissue culture. So there's the yield, it's maybe water weight. You can put on more water weight, but that's not true yield. If you want to get at about 10%, 9% water weight, activity water level is what I like my medical cannabis to be at. Get it around to that. You'll find that the yield won't vary. You can leave it for a longer time, and let it foxtail, because that's growing more, but that'll take days. So by stressing the plant out, that's only going to activate its immune pathways to try and fix those stress problems in the plant, like when we're sick. So yeah, yield. The techniques in yield take a few weeks earlier, or you can grow them a bit longer. You can put cytokinins back into them and reverse the extend the flowering cycle. I've done that up to 60-day cultivars, up to 105 days. I've done that before. I've done a pretty good video on cannabis senescence, which shows you how to extend your life of your plants. You can, by using various hormones, quite a few hormones I go through on that. It's very extensive, that video. All right, looking down. Hey, Stony Creek, how you going, mate? Appreciate your help and learning. Good on you. Thanks for your nice feedback and words. Thanks for that, CC. Oh, good on you. Oh, you nailed it. Hey, sweet. I like to hear that. Family Falk says, does anybody know anything about isomerizing and CBD converting into THC under certain circumstances? Yes, it does happen. It's synthetic, though. This is the thing. All that they're doing, they're adding a tail onto it. You're in the States, and in Australia, it's not. Actually, we got medical extraction, but they don't do it. But anyway, that's how it happens. It's synthetic, but remember, synthetic cannabis has been banned in a few, Oregon, and a few other places in the States. They've banned it. So it is possible and it is done, but that's how you do it. It's to do with the tail. I suppose I can show you a chart. Why not? I haven't shown many today. What would that be under? Chromosomes it would be under. Chromosome mutations, possibly. Let's have a look. I'll share my screen, show you all. Where is it? This one. Share. So I'm looking for the molecular structure of THC. That's what I'm showing you now. That's the charts. Triploid. Oh, actually, no. I'm not going to go into it. I'm going to try and just show. Stick to the question. And diploidy mutations. No. I'm trying to look for fragile sites, gene mutations, naval, spontaneous, induced mutations. Yes, there's quite a few different ones. I might stick on topic. That's why we have a lot of Australia, because they come in when they get zapped, they get mutated. It is a mutagen. They're X-rays. Frame shift. That's cool. I don't know. Get back to it. Applications. Polyploids. No. Might just have to. Yeah. I'm sure I've got it though. I really want to show you. I'll try again. Share screen. Chromosome mutations. Come on, where is it? Maybe it's in secondary metabolites, unless it's down here. No, it's a big long tail thing. Yes. It says, actually, I've even called it chop the tail, because that's how you change the different molecular structure of it. So I've even worded it as that, but I don't know how to search my computer for a file called chop the tail. Isolate flavors. It might be boiling points. No, trichomes, flavonoids, terpenes. Let's have a look. No. There's some tails. There's some. All right, let's have a look in here. Anti-cancer terpenes. What a way to pull it up. All right, we'll just have a look at this. So some of the anti-cancer terpenes. So if you were in your cultivar, you would be hoping that you want to get palitaxel. That's one of the terpenes that's anti-cancer. Sarcodityn, sarcodityn. Oh gosh, look at that big long one. Conimig, sterol, Continag, sterol, and squalarmine. So if you want to, you combine your THC, CBD with these as well. There's some down the bottom. I can't really see them. But what they do with the THC molecule is, say if it's this long, like in this one here, or we'll go up to the top one. That's pretty easy. They're parallel alcohol. So that's also an anti-cancer terpenoid. So if it's really long, that's what THC is. You chop the tail off a bit by adding different chemicals, and it'll change it. And then that will change it into a different molecule. And that's what the molecule would then be, according to its size and shape, it would be renamed. And then that's how they do it. Cool. I didn't show you the one specific, but this actually relates to today, so it's actually a bit better. I hope that helps, Family Falc. So yeah, just be careful about the synthetics about it, mate, because they did have to use chemicals to alter it. It's not natural. Good question, though. Very related today. I just meant, has anybody tried it and seen it? Oh, I don't know what's that for. Hospice is Oh, death care. Oh, we call that palliative care in Australia. So he's on hospice. Oh, so in other words, he needs the hard cores. So yeah, it'd be extracting it for him. I'd be definitely extracting it for him, mate. I'd be even double distilling it, or you could, about not fermentation, about isolate, where you're going to put your glass beaker and boil it off, and it goes to fumes, and then you catch the fumes, and it goes into your second beaker. You've probably got them. I reckon you have GMF. So that's another way. But I would, jam as much as you can into your, the different extraction ways. I'd try and get the strongest extraction I can for that person, because they need very strong things in palliative care, because a lot of stuff doesn't really work because they're at the end of their life and their body's shutting down. So you want to help them activate all their pain, shut their pathways for them so they can have some sort of quality of life towards the end. Good on you, mate, for trying. Thanks for Big Aussie Dave for helping me with my words. Mick, wow, strange burning smell. Okay. Basically is what we're comfortable. Yes. Oh, yeah. Hospice means stopping treatments for his conditions. Oh, he's living out of his days. Oh, really? They stop. So it's like a good luck? That's worse than palliative care. That looks like a bloody good luck. Oh, mate. Good on you for blimmin' trying. Yeah. Any people, I don't want to say that. It'd be good to shout you out where you are, mate. So, it's, so sad. So good on, at least he's trying. So if you know of any other people that have had that type of an attitude, have been to hospitals that just said good luck, and all of their other treatments aren't working, try and look at some strong cannabinoids with a combination of those different terpenes, if it's cancer, for instance. Definitely. All right. Yes, Big Aussie Dave. He is at peace eventually. Sorry to hear it, GMF. Yo, GMF, cheers, mate. It's a simple conversion of an acid at temperature. Oh, yeah, so they chop the tail, they use acid at a temperature. Very easy, yes. You're just changing, yes. THC, thanks, bud. Oh, some good positive words getting thrown around the chat. Nice to see everybody. Mick, it's not burning hair, is it? You're burning your hair, mate. That's funny. AussyCC, that was hilarious, but you are right. Quicker drying is good for mold. Oh, that's what I was up to before. Thank you. I get shit for drying in jars 4 days, but it works well. Mate, thanks. From post-harvest productions at agricultural class, I was speaking with a professor afterwards about different ways, novel ways where we can do it for medical cannabis. And he said, microwave-assisted vacuuming is a way that it'll work with us. And what's it called? There's a fancy word for it, but if it's infrared drying. So you use infrared radiation. And the reason why these two techniques are very good, because it gets the temperature below 50 Celsius or about 140 Fahrenheit. And that's because that's when our terpenes start at. So they're volatile at those temperatures. So if you were to do an extraction or a cook above those temperatures, you will not have CBG and OC mean in your result, because it will have volatilized off. So these are two techniques where we can do it under that. My technique of doing it in the oven, I've got some fast ways of doing it. It's been patented by Germany now, and it's similar sciences behind it as well. I can successfully do it in a few days, maintaining all of the terpenes and cannabinoids. And GMF's done it for a while too. And yeah, people, it's because they just don't know education, mate. They haven't tried it. They hear too much bro science about it. It can't work. It can't. Well, if you get your science into it, it can. Both you and me have done it. It's cool. There's all these good different ways where people can learn and try, and it's going to suit their system. That's what I was getting to about that competition. So by these new novel ways, by introducing them into people's grows and their techniques, they can get better results, and there's chance for fungus and mold is virtually nil, because every time it's hanging there, we've got spores that are in the air. You're not hanging them in lab conditions that are fully HEPA filtered hardcore where you're drying at. So there's always spores in the air of some type. So by drying it quicker, you lessen the chance of it holding its medicinal quality and not having those spores on it. Brad, thanks GMF for helping me get my words out. Vin asks, are there any bad reactions with pharmaceuticals and cannabis? Well, from my experience, yeah, people take too many pharmaceuticals and not enough cannabis. So when I was in the dispensaries, I had a ACMPO license. I was working under one of them in Canada and selling to the dispensaries. I would ask the oldies when I was in there, why are you in here? And they'd say, because I can take heaps of capsules or tablets, or I can just take a little bit of cannabis oil or cannabis extract or something along those lines. And so I definitely know that it works. So, yeah, that's why I really want to, that's why it promotes me to study it and to get into it more heavily, because it's a drug that is so beneficial for the community. It's just that big pharma, and they can't make money off it, because that's why they'll pay the doctors, and the doctors will get kicked. Actually, that's why it is where it is at the moment. And Australia is not, I don't want to get too political, but are there any bad reactions, Vin asks, with pharmaceuticals and cannabis? That's my answer, is people take too much pharmaceuticals and not enough cannabis. Everybody's got a different condition and reason why, though. So, and different strength. A lot of cannabis, especially in Australia, is pretty crap. Even the medical stuff's pretty crap. It's getting better. It does have terpenes in it now, meaning it does open up that avenue to give the entourage effect, where if there's no smell into it, you're really limiting just the cannabinoids are going to do its trick for you. So it depends what your medicine genetics are and how it's handled after it's been harvested. Good question, Vin. Hey, Cool Hand Luke, how you going, mate? Hi, Stony Creek says highest humidity possible without causing mold helps to know your cultivar when drying, I reckon. Yes. Saccharomyces cerevisiae has an activity water level of 65, meaning at 65% humidity, it can germinate or sprout. So it's really low. But of course, not everyone has Saccharomyces in their air, but it just goes to show that it can happen at very low. I'll put that up. Oh, no. It is up. Okay. Stony Creek, morning, Luke. Shame Nexus isn't here. Oh, well. Can't do nothing yet. It doesn't, mate. Good. Oh, sweet. It does help. That's really good. Cool, and Luke. Seeing the THC molecule as a tattoo. And look who's next. That's pretty funny. Hope you're getting a laugh as well, mate. That's funny. G'day, mate. That's pretty cool. It would make a good tattoo, mate. And that's probably something I should think about. I've got to, actually, I'm not going to talk about that. GMF says, concentrates it is then. Thanks, AussyCC. Yeah, mate, you're welcome. That'll help the palliative cancer patients and the life patients. Poor people, they need as much as they can get, some sort of quality. And even if it helps them sleep, you know, they're still going to sleep soundly. It's, yeah, because when you have too much of it, usually you just sleep. That's what I do. Like when I was in Canada, I had my oil, and if you do an extraction, you'll know how much do I put in this gummy. So I had, say, 2 mils. It did something. I had 3 mils the following day. It put me to sleep. So I know 3 mils was my limit. So in each gummy, I only want to put in 2 mils, for instance. So if you're a friend, possibly you could do a test like that, know how high it is. So you'd say to him, if you take 2 of these, it'll put you to sleep. If you take 1, it might just kill a lot of your pain, as an example. I'm thinking about making vape shoot. Okay. Very good. Very good, mate. Hey, Koski. How you going, girl, Koski? Nice to see you. All right, Koski, I've known you for some years now. Nearly, bloody hell, it's getting on, isn't it? Because Dave, middle of last decade, I think I met that fella. Oh, yep, the drying process. Oh, yeah, Aussie Auto says he likes a 2-week slow dry. Yep. Monty says he likes a 10 to 14 day process. I used to like about a 10-day process too. I used to book the trimmers about day 10-ish, and in Canada that is, and it's used to be good. But now I've got a faster way that gets the better result, that they don't have to be hanging around, possibly germinating, releasing all their ethylene, trying to keep the cells slowly dying. Because as you know, when you harvest, the cell's still alive. It's releasing gases, it increases CO2 and ethylene, and it's finishing off. So by halting that process, you can also get all those terpenes that are gathered at that point as well. Yes, Gokowski, how you going? Says Main Green. Lene says, I've personally never had any bad interaction with cannabis and various pharmaceuticals. There you go. There's some first-hand experience. Aussie Autos, Monty. Oh, we're down to the bottom. Koski likes a 10-day dry. Yep. Aussie Autos says, they say after a good slow dry, the cannabis is cured if done right. Yeah, okay. That's what they say, is it? Yeah, well, depends on the cultivar. I've smoked some straight away from drying, and it can get you just as high as it would after waiting 2 weeks in an airtight container by curing it. And Death Bubba, for instance, that was as soon as it's harvested, straight away as it's dried, you can smoke it, and it's going to get you just as affected as it would as if it was 2 or 3 week cured. And then Gorilla Glue, it's the opposite for me. When I grew it and dried it, it didn't do much, and I was a bit upset. I waited 10 days. After 10 days, it was holy mackerel. Wow. And it turned out to be still to this day one of the strongest I've smoked. And at the dispensary twice, it broke their machine when it went over 30 for THC. But they do say that curing does definitely help. The dispensary too with curing, they didn't want me to have trichomes that were too amber, because that used to fasten the ripening process. So it would expire faster. The shelf life would be lower. So they wanted me to stuff with just a little bit of amber trikes, just getting into it. So that's how I used to pick it for them. But I like to pick mine generally with probably at least 30% amber trikes, because I know mine's all going to get smoked within 8 weeks. All right, we're nearly down to it. No questions. Aussie Auto says, I still always cure, but after a good slow dry on my wetting glue, was ready to go. Hey, that's the way. Very good. All right, well, it's only been an hour. Full on. It's been good. Thank you, World Cancer Day. It was really good. Oh, here's someone new. Hello, Mr. FUCR. How are you, mate? Cheers, everybody, he says. That's the way. You just missed it right at the end, mate. Lucky it's a recorded show and you can re-watch it. And, oh, that's right, for that little cash button underneath, if anybody's got any persons there, I'll pull up that slide, actually, because I haven't said it for quite a few weeks, and I've got to mention it for the monetarizations stuff. Where is it? There's 4 different things down the bottom. Lives, monetarization, diversity. I'm going to have to look for it. I don't want to do it from the top of my head. I've done it for a while. Is that it? Sweet, here it is. I found it. Stop looking, everybody. I found it. Share screen. So there's 4 different things that I've set up for the monetarization. Oh, Jesus. There it is. $5 is for support. Thank you. $80 gets my private lectures. I've got a different list that I haven't put in there of a few botany ones, and there's about a dozen. And $150 a month gets a 120-minute call and a novelty gift like a chart or something that I've made. And $300 gets a weekly call for like a company starting up or a person going to the doctors, medical cannabis doctors or something like that that I can give my experience or education with. So that's, if you want to help out, that's that's what the cash button underneath down the bottom does. Thank you to everybody who was there's been a few people that have done that. So I really appreciate your help, guys and girls, for that. It's good. And that's it. So next week, it'll be another open medical cannabis topic. So if you'd like to bring your questions, today was a really good one. I think we went on with a bit of drying stuff. I'll maybe relabel it from the cancer thing. I'll maybe try and slip in drying as well, because I think we went over a few different techniques and a few different people mentioned their ways. I mentioned a few different novel ways which are coming up in the future. And yeah, so it was good. Appreciate everybody for turning up in the chat and asking your questions. Thanks heaps again. So I'll just, there's a few things clicking through here. Lene, thank you, good show. Bananarama, Cruel Hand, peace out. Saturate yourselves with cannabinoids to stay healthy and cancer free. That's the way. Cool, freeze. Yeah, peace out. Come on, Luke. Hey, thanks, brother. Good on you, mate. Cheers for your good words. Jezza Chronic, just on the way out. You've missed it, mate. Anyone want to pay me not to be a NPC? Don't know what that is. Don't know. Put your effort in, Jezza. You'll get there, mate. Vinny says thanks. Good on you, mate. Thank you again. For the best, you did great. Share the knowledge in a clearly spoken, non-paranoid event. Thanks to chat. Yes, good on you, chat. Keeping the drama low. It's nice. It's all about cannabis, medical cannabis around here. Very good. Aussie Autos, Jeff Palia. Been lurking and working. Yeah, you're always welcome, Jeff. Like anybody else is always welcome. Even the people who have problems and want to spam and things, because they've got problems, so we can help them with some sort of medical cannabis. And I'm sure they'll be much appreciated afterwards. They're relaxed. So thanks everybody again, and I'll see you next week again for another open medical cannabis topic. Bring your questions and have a good week. So it's good breed, happy breeding. Oh, if I forget my little spiel. Happy breeding, happy growing, and good health to you all. Thank you. That's it. Bye-bye. Yay.